This is Chapter 3, Module 1, Cytoplasm and Organelles of the Animal Cell. The learning objectives of this module are 1. Define cytoplasm and cytosol and describe its composition. 2. Describe the structure, function, and relationship of all membranous cell organelles. And 3. Describe the structure, function, and relationship of all non-membranous cell organelles. A scientist named Robert Hooke developed a cell theory that states that all cells are the building blocks of all plants and animals. His theory states that all cells come from the division of pre-existing cells, and that they are the smallest units that perform all the vital physiological functions. In other words, a cell must reproduce to form another cell, and each cell maintains homeostasis on its own. In the human body, some cells are sex cells or reproductive cells, such as the sperm in the male or the egg in the female. But the sex cells only make up a small fraction of the cells in the human body. We will discuss sex cells when we look at the reproductive system. Most of the cells found in the human body are somatic cells. Soma is another word for body, so somatic cells include all body cells except for sex cells. The contents of the cell are enclosed within a cell membrane. The watery fluid outside of the cell is called the extracellular fluid. Another name for the extracellular fluid is interstitial fluid. Think in the tissue fluid. The fluid inside the cell is called the cytosol, or otherwise known as intracellular fluid. The interstitial fluid and the cytosol are separated by the cell membrane, also called the plasma membrane. The cytosol contains many dissolved materials such as nutrients, ions, proteins, and waste products in varying amounts. For example, within the cytosol are high ion concentrations of potassium, but low concentrations of sodium. The cytosol also contains high concentrations of protein and carbohydrates and smaller amounts of amino acids and fats. In contrast, the interstitial fluid or extracellular fluid contains a high concentration of sodium, but a low concentration of potassium, and very few proteins are found in the extracellular fluid. The concentration differences of sodium and potassium are very important in describing the cell's membrane potential, which will be discussed in a different module. The cytoplasm is all of the contents found within the cell. The cytoplasm contains several organelles. Organelle means tiny organ, so essentially the organelles are the cell's organs. Some of these organelles are membranous, and some of them are non-membranous. Membranous organelles have a membrane mostly made up of phospholipids that completely surround the organelle. Non-membranous organelles either completely lack this membrane or they are only partially covered by a phospholipid membrane. The membranous organelles include the endoplasmic reticulum, the Golgi apparatus, the mitochondria, the peroxisomes, the lysosomes, and the nucleus. The nucleus is the largest of all the organelles. The endoplasmic reticulum is a network within the cell that contains storage chambers and channels inside a membrane that is similar to the cell membrane. The endoplasmic reticulum makes or synthesizes our basic organic molecules such as proteins, carbohydrates, and lipids that our cells can use. The endoplasmic reticulum can also store these molecules and transport them throughout the endoplasmic reticulum to other areas of the cell. The endoplasmic reticulum also helps to detoxify drugs or toxins entering the cell. The endoplasmic reticulum can either be smooth or rough. The smooth endoplasmic reticulum doesn't have any ribosomes attached to it. It synthesizes lipids and carbohydrates that can be used for various functions in the body. Phospholipids and cholesterol are lipids that will be used to repair the cell membrane. Steroid hormones will be used in the reproductive system. Glycerides and glycogen are synthesized as molecules that store fats and carbohydrates in the liver, muscles, and fat cells. The rough endoplasmic reticulum is called rough because its surface is covered with ribosomes, making it bumpy or rough. The rough endoplasmic reticulum synthesizes protein and glycoprotein products that will be modified into different protein shapes and structures and then enclosed in vesicles. These vesicles are called transport vesicles. They will pinch off of the endoplasmic reticulum and transport those proteins either to the inside of the cell or to the Golgi apparatus. The Golgi apparatus looks like stacks of dishes. They are actually stacks of flattened membranes called cisternae. The Golgi apparatus is sort of a packaging plant, and it has three basic functions. First, it modifies and packages secretions, such as hormones or enzymes, into small vesicles that will be released from the cell into the extracellular fluid through exocytosis. The Golgi apparatus also renews or modifies the plasma membrane. It does this by forming a vesicle that fuses with the cell membrane. Since the membrane of the vesicle is the same phospholipid bilayer membrane of the cell, the two membranes become seamlessly joined. This is a necessary maintenance, as the cell membrane gets worn down or damaged and needs to be replaced. And third, the Golgi apparatus packages special enzymes within vesicles for use in the cytoplasm. To look at how the endoplasmic reticulum and the Golgi apparatus work together, we will consider proteins. A protein is produced at the ribosome on a rough endoplasmic reticulum. From the ribosome, the protein enters into the chambers of the endoplasmic reticulum and becomes modified. The protein may change shape into a tertiary or quaternary structure. Then the protein is transported through the chambers of the endoplasmic reticulum to the outer reaches of the cell. At the end of the endoplasmic reticulum, the protein will be enclosed in a transport vesicle as the membrane of the endoplasmic reticulum pinches off and creates this vesicle. Much like a vesicle in a lava lamp is formed that floats to the surface of the lamp. This vesicle then floats to the Golgi apparatus, which is closer to the surface of the cell. The Golgi apparatus creates secretory vesicles which are released toward the cell membrane. These secretory vesicles fuse with the cell membrane and release the protein out into the extracellular fluid. Now this protein can be used in other places in the body. Maybe the protein is an enzyme that's needed to catalyze reactions somewhere else in the body. Maybe this protein is a hormone that will travel to other places in the body. Either way, the cell has created a protein which is now released outside of the cell. The mitochondria is an organelle that has two membranes. The inner membrane has numerous folds in it called cristae. There is a fluid in between the outer membrane and the inner membrane. There's also a fluid inside the inner membrane. The fluid inside the inner membrane is called the matrix. The mitochondrion uses glucose from the food that we eat to produce energy, which is called ATP. Three processes are required in order for ATP to be produced by the mitochondrion: glycolysis, which occurs in the cytoplasm of the cell, the citric acid, which occurs in the matrix of the mitochondrion, and the electron transport system, which occurs in the inner mitochondrial membrane. More of these systems will be discussed in our module on metabolism. Peroxisomes are vesicles surrounded by a membrane. They contain enzymes that break down fatty acids and other organic compounds. As they do so, they produce hydrogen peroxide, or H2O2, which is very toxic to the body. So hydrogen peroxide will have to be broken down into water and oxygen. Peroxisomes come from other peroxisomes, unlike the lysosome, which comes from the Golgi apparatus. The lysosome's job is to clean up the inside of the cells. The lysosome is a membrane-covered vesicle that came from the Golgi apparatus. It is filled with digestive enzymes. These lysosomes can attack bacteria, recycle damaged organelles, and break down large molecules. Once these lysosomes digest these things, they will eject them from the cell through exocytosis. When cells are damaged beyond repair, or when they have aged to the point they are no longer functional, the lysosomes will begin to deteriorate. The lysosomal membrane breaks down and digestive enzymes are released. These enzymes will eat away at the entire cell, causing it to decompose. Some of the cellular materials will recycle and be used by other cells, but the cell itself has self-destructed. This self-destruction of damaged cells is called autolysis, or apoptosis. The nucleus is the largest organelle and is the cell's control center. It has a membrane that is called the nuclear envelope that is a double membrane surrounding the nucleus. Between the two membranes is a space called the perinuclear space. Several openings in the nuclear membranes, called the nuclear pores, act as passageways for specific molecules, such as proteins, to pass through. The fluid within the nucleus is called the nucleoplasm, which contains ions, enzymes, nucleotides, and some RNA. The nucleus also contains DNA, which is the molecule that carries the information needed for the cell to survive. If the cell is not dividing, the DNA will be loosely coiled and is called chromatin that looks like fine thread within the nucleus. If the cell is preparing to divide, the DNA become tightly coiled and are called chromosomes. DNA coils itself around proteins called histones that help it to tightly coil when the cell is dividing. Other important structures within the nucleus are the nucleolus, or nucleoli for plural, that makes the small and large ribosomal units, and also synthesizes ribosomal RNA. The non-membranous organelles include the cytoskeleton, the centrioles, the microvilli, cilia, the proteasomes, and the ribosomes. The cytoskeleton is an important part of the cell's ability to survive. The cytoskeleton contains 4 main structural proteins with various functions: the microfilaments, the intermediate filaments, the microtubules, and the thick filaments. Microfilaments are thin filaments that contain a protein called actin. These microfilaments provide mechanical strength for the cell, and they give the cytoplasm a thicker consistency. Intermediate filaments are mid-sized filaments that give the cell its durability and strength. The collagen fibers of the intermediate fibers allow the cell to be distorted and then return to its original shape. The intermediate fibers also hold the organelles in place to stabilize them. They can also stabilize a cell's position within its environment by binding to integral proteins in the cell membrane that are bound to extracellular structures. Microtubules are large, hollow tubes that have several functions. They are attached to the centrosome, which is a small area near the nucleus, and they extend out toward the cell membrane, kind of like the spokes of a wheel. Organelles will anchor themselves to these tubules and move around the cells. Vesicles can move around the cell as well through the hollow center of these tubules. When the cell begins to divide into 2 cells, these microtubules will arrange themselves into a football-shaped structure called the spindle apparatus that extends from one end of the cell to the other. Thick filaments are the thickest of the cytoskeleton components. They contain a protein called myosin. Thick filaments are only found in muscle cells, and they interact with actin and the thin filaments to provide muscle contraction. Non-membranous organelles other than cytoskeleton include the microvilli, cilia, and centrioles. Microvilli are extensions of the cell membrane that protrude away from the cell like fingers. The purpose of the microvilli is to increase the surface area of the cell. Increasing the surface area of the cell gives more area for things to be absorbed into the cell. The cytoskeleton helps to give these microvilli their shape. Microvilli are found in the cells of the digestive tract where nutrients that you eat will be absorbed into the cells. Cilia are thinner extensions that look like small hairs protruding from the cell. They are also formed by microtubules that push the cell membrane outward. The cilia move in a rhythmic, vacillating motion to help move substances across the surface of the cell. The cilia are found in the respiratory tract, where cilia will help to move mucus and dust up and along the air passageways to the throat, where the mucus can then be swallowed or spit out. The centrioles are small structures that are made of small microtubules. Each cell that contains a nucleus will have a pair of centrioles. They are found in an area of cytoplasm near the nucleus called the centrosome. Remember, this is the same area that the microtubules extend from. The centrioles will replicate to produce 2 pairs of centrioles during cell division, and they will control the construction of the spindle apparatus during that time. Not all of the cells in the body have the same organelles or the same number of organelles, because different cell types have different shapes and different functions. This ends Module 1 of Chapter 3, Cytoplasm and Organelles of the Animal Cell.